please protect me. He will try to find out the solution, okay, or try to reduce your punishment at least. And the third thing we can add, to be honest and be, other way you can say truth to your teacher, that yes sir, I have not understand what you are telling. Feel free, this is not a big crime. If you are not understand, yes, it may be, it's possible. It's not everyone is all right, okay, or equal, no. And even if you can ask many questions, I can answer. Not possible to know everything, but what little we know, that should be very clear, that yes sir, I know this much only, and beyond this I don't know. It's okay, we can learn, right? We can learn from others, we can discuss among ourselves to clear that doubt, okay? So yeah, please.
Sir,
okay.
Your video will be covered, so we need white space here slightly. So
oh, I should be here this side? Oh, I should be here only, but I wish to move. I love moving here and there, going to the student. It's okay. Okay, for this, okay, this gap is required. Oh, I should be very careful. I can't be careful. Like, I'm feeling like an actor. Yeah, that's okay. Thank you. So, friends, our topic of discussion will be fundamentals of Qtl mapping. Fundamentals means we will discuss the very basic things. I know many of you already know is better, but anyway, let us brush it up, okay? And if anything we have in our mind of doubt, perhaps we can discuss and clarify those things again. Is it okay? Right. Okay. Now, before going depth into the topic, we have to know each and every words. It matters a lot. So, fundamentals of Qtl mapping. So, first we must know what is Qtl. Before Qtl, again there are two things, Qt. First we should be clear with Qt, then we can add L to make it Qtl. Qt have two meanings. One is your quantitative trait, other one is your qualitative trait. Okay, we know as that. And most of the properties you already have studied, starting from your undergrads, then master, and some of you are already studying the things in case of your PhD program, right? Okay, the first thing they said, in case of qualitative characters, so they are governed by single or a few genes, right? In contrast to that, quantitative characters are governed by many genes. That's a simple difference, you know about that. Qualitative character shows a discrete classes. I will show what type of discrete class we mean. So, it shows this discrete classes, but in case of quantitative characters, it does not show that discrete class, but it shows a continuous type of variation, right? Continuous, no this clear cut difference. So, there is a merging of classes. So, ultimately we get a continuous type of classes. Other difference you see, easy to transfer through hybridization, which you can say difficult in case of quantitative character. Can I ask you why it should be difficult? You said in case of qualitative character, you said easy to transfer through hybridization. So, what that quantitative character has the problem? Why it cannot be transferred easily? By many genes, one discontinuous variation. It shows continuous variation. There are a number of genes. Any other problem? Highly influenced by the environment. Population size is a matter. Any other factors coming up to your mind? Feel free to think. Minor genes. Minor genes what? Governed by minor genes. Linkage drag problem. Any other point coming to your mind? Feel free to think. Phenotyping is very difficult. Why? It is having a continuous type of variations. Okay. So, whatever you have, you just what the points coming to your mind, almost all of them are correct. That yes, in case of major gene, it is located in a particular place in the genome. So, through hybridization, you can transfer that particular region to your target genome, right? But since she is mentioned rightly, it is governed by so many genes, located in so different places. So at one go, perhaps it's not possible to transfer all those things together, right? Had it been this lock, I perhaps can transfer easily. One is here, maybe one somewhere, one the back side, the other this side, the other. So in one crossing, because crossing is what? Again, it's coming through only on your recombination, through crossing over. Is exchange of segments, right? Then that is the way you are transferring the genes and QTLs. So that makes a huge difference between this qualitative and quantitative characters. Yeah. Finally, what do you say? Yeah. The first one is less influenced by the environment. The other one was highly influenced by the environment because its individual effect is very, very low. That's why it's also influenced by the environment. Okay, fine. So that's why I said that this is your discrete class. If you talk about discrete class, so you can show that yes, this is my discrete class. The left side you can see here red and white. If it is red, it is red. If it is white, it is white. No intermediate, no pink here, right? But in that case, you see that yield. Somewhere it is say 10 grams, some 20, 30, 35, 40. It may go beyond that way. So there is a continuous type of variation. If I take plant height of the student present here, almost same thing will happen. Some will say 160, some 165, one may be 75, 170. Also that way, not a clear that yes, all are will be 100, rest will be 150. Not like that. All kinds of will be there. Okay, so those are specific characteristics of your quantitative characters, right? So we have now understood about the QT, quantitative trait and qualitative trait. Then add to this is your loci, plural of locus, right? All right. So now then how do you define that QTL, quantitative trait loci? Now here you see this term was first used in 1975 given by Gilderman. Now it basically represents, it has been written here, it represents a genomic region that is responsible for controlling a specific trait. You are not talking about a single gene, you are talking about a genomic region. So that difference should be in your mind in future, because if you keep this word in mind, many questions you will be able to answer. Why there is so much variation? This variation lies here because you are talking about a genomic region, means a segment. You are not talking a single or particular gene, but in that segment there may be many more genes, many genes, maybe for same character, maybe for different character, but you are talking of the whole segment as a whole, right? So that's why you are calling it as a what is QTL? As a genomic region, a segment of the chromosome, segment of the chromosome. That segment of the chromosome is responsible for a particular character, right? Remember, it may contain one or many genes. Okay, we will come to this again, all right? Yeah, that has been mentioned about this cluster of linked genes. Cluster of linked genes. Can anybody tell me what you mean by this linked genes and unlinked genes? She will tell us. Inherited together, right? So should they be located also close by, or they can located elsewhere? Very close by, right? Any other opinion about this linked gene? She said that they should be located close by and inherited together. Any other opinion about this linkage? Yeah, please be free, feel free. I said feel free, no problem. I am not going to deduct your marks.
So linked gene must be present on the same chromosome.
Must be the same chromosome. Is it so? Is it universal? Why not? Who says no? Let us debate. Let us debate. You are at that stage. I always say in the class, you are at that stage now to debate. It's not necessary anyone will say and you will accept it. You must have your own opinion.
On the two arms,
Yeah.
and having this distance more than 50 centimorgan, segregate independently. We are not saying the genes present on the same chromosome are not to be linked.
Not to be linked. Okay, perhaps we will somewhere mention about association mapping, right? Linkage disequilibrium concept is used there. Name of the technique is your association mapping. So there perhaps you have the other opinion also, right? That what he is saying that it should be close, somebody has rightly said they are inherited together, right? Yeah, that everything is very perfectly fine. Also should remember that what happened in case of other techniques. Association mapping is a very popular technique now. So what happens there about the linkage? Perhaps there is something differences also. We will have to come out. All right, fine. Yes, that I already mentioned that in case of QTL, not necessary they should be located in one chromosome because it is governed by many genes. Not necessary all the genes will be sitting together like you. They may be in different parts of the genome. All right, these are different properties. So remember, you are talking about a segment of chromosome that is controlling a particular target, quantitative trait. What is the principle? If we understand the principle, rest of the approaches will be very easy for us to understand. Principle in a very crude sense I am talking about, very crude sense. What we do here, first you make genotype of the mapping population, then you group the plants as per their genotype. Say for example, here I am giving example of A and A. I think we have, yeah, one table we have, we can see that also. So first what you will do the genotyping and group the plants based on the genotype. You remember, we are now targeting only two genotypes, A, capital A and small a, because most often what you use is your homozygous population. You are talking about real for quality QTL mapping. So when you use that real, it is by that time already homozygous. Maybe one or two locus is there still heterozygous, but mostly you will get it in homozygous condition. Okay, question may comes. Some of my friends may say, sir, I am using F2 population. What will happen to me? Because F2 will have three different types of genotypes. There will be one more class, capital A, small a. So what will happen to that, right? But perhaps we are now making class only with these two groups, capital A, small a. All right, fine. Based on that, you analyze the phenotypic means of these two groups. Based on the genotype, you arrange the data. All capital A data should be together, and all small a plants, their data should be together. Now you have got two group of data. Then you see what is the difference between these two, and is it statistically significant? There may be one or two difference, but that must be statistically significant. If that difference is statistically significant, then that particular marker based on which you are classifying, that marker is linked to that particular character. Let us see that example, then it will be easy for us. Say for example, these are my 10 plants. 10 plants. I have genotype with the marker M. Okay, the marker M. So this plant number 1 genotype is capital A. Plant number 2 is small a, and so on. Okay. Now, based on and accordingly you have the phenotypic data. That if this is yield, so for plant number 1, 45 gram is your yield. Plant number 2, 42 gram is your yield. Okay, that way you have the yield data. What I am telling in our principle, what you say that first I have to make them group. So I will make a group of all AA plants. AA 45, next AA is 30, next AA is 45, is it? Next AA is 60, next AA is 47. So out of this 10, these are the plants. How many plants are there? 6 plants. So they are having genotype capital A. You take them and make it a total. Similarly, you take the other genotype and take out their total. Is it okay? Now you see, I have got for AA 259, for small a, I have got 200 218. Now this difference 259 and 218, is it statistically significant? I have to use maybe f-trace, maybe t-test. So if you have two groups, so you are using the t-test. So in that t-test, if that is significant, then you say the marker M, because this classification is for what? Marker M. So this marker M is linked with E, the particular character. If it is for plant height, marker number N, so you will say marker number N is linked with character plant height. So whatever be the character. So this is the basic principle of your QTL mapping. Is it okay now? What you will do first? Genotypically divide the plants, then take their phenotypic data, club together group wise, see the statistical significance. If they are statistically significant, you say that particular marker is linked with that QTL for the character. If it is not significant, then you say this marker is not linked with this particular character. Is it okay now? Yeah, yeah.
In particular crop, marker markers are not available means how we can say whether it is.
Without that, you cannot do that. QTL analysis. Earlier we have other concepts, biometrical approaches, we have there, but that is not your QTL mapping. Okay, but here I will show you the requirements, then you will realize that whether you can use for your purpose. Suppose this is your situation, yes, this is good. My situation is like this, I have working on a crop, that particular crop is having no molecular marker. Can I go for QTL analysis? Right? That is her question. That we will see the answer next in a few slides away from here. What is needed? You see the answer here. What is needed here? You first need to have one mapping population. I am not sure today morning I think that mapping population was covered. No, then there will be another lecture exclusively for mapping population. But anyway, let me mention, and most of you perhaps know about that. So you need a mapping population. Can you define what is mapping population? What about your basic knowledge about mapping population?
It's a group of genotype which used for mapping of gene. There is a various procedures for developing the mapping population, like back crosses. F1 is continuously back cross, we call it PIL. Or
Second, can you tell me what you think a mapping population? I have a collection of plants. Can I use it as a mapping population?
so,
You will answer. Yeah.
so mapping population is basically we say biparental mapping population.
Only.
No sir, germ class lines can also be used as a mapping population, but that's a different aspect in case of
Correct.
association mapping and all. Biparental mapping population is only used for like QTL linkage mapping.
The third one, what do you think? What is called mapping population?
Populations like different populations which we can be mapped by using different procedures.
Amresh, what is mapping population?
Population which are derived from the crossing between two diverse parents, should be F2, F3, and there is double applets, immortalized, mortalized.
What is mapping population?
Population having segregation of two different alleles of a particular trait.
Any other opinion about the mapping population?
It is a
Population which is developed by crossing a diverse genotype which exhibit the segregation pattern of the trait of interest.
She was mentioning that if I have not a biparental mapping population, also I can use that as a mapping population. Germplasm. She said that I can use germplasm as my mapping population. Can I? We can. So then in that case, okay, I agree, you can. So in that case, your definition should not be that only it is biparental or something like. Yeah, please.
it includes biparental or germplasm.
Many parental also.
Mapping a particular gene of interest or a group of.
Can we have many parental mapping population?
Yes, sir.
We have. Magic is a many parental, right? Yeah. NAM is a many parents, right? Okay. So in that case, I think again we have to verify that what should be the definition. All right. I think there will be a special class on that. After that, we can discuss again. All right. Oh, fine. So you need a mapping population. So we said that what most ideal, DH real. There is a question mark in F2. Why? Why we are putting a question mark in F2? Can I not use that F2 as a mapping population? Yeah, please, there is the last one. Yeah, not, yes, you tell me. F2, have you heard that F2 can also be a mapping population?
Can be used, but some limitations are there. Only one periodic cycle we would have.
But we can use, right? Any doubt about this? Okay, another question about this mapping population F2. In case of real, we say we need to replicate, we need to grow it over location, such and such thing. Can we do in case of F2? Why not? Not IRI student, not IRI student. Let us discuss, okay, get the opinion from outside also. Yeah, why you cannot replicate F2? So what? So what? Each and every plant is different. So what? What is, why you think, yes, okay, this is your opinion. He said that all plants are different, so I cannot repeat. What is the problem?
It's a segregating population.
So what?
It gets many combinations and it's not settled down.
What you think? Why F2 cannot be repeated, means replicated?
Yes, suitable only one cycle of recombination and 1:1 ratios.
Uh-huh.
We can't replicate. Means number of plants are available.
Okay, first in that case, if you think you should first learn the meaning of replication. You know meaning of replication? Repetition. Repetition means what? Same thing should be done. He has rightly mentioned my all plants are different. Suppose I have 100 F2 plants. 100 plants all are different. If I divide it 50 in New Delhi, 50 in Kerala, so they are different F2. But that meaning of replication, replicated trial is what? Same 50 plants should be grown here. Seeds of those same 50 plants should be grown there. So then only it is called replication. But in this case, you are not doing that. He is right that my each and every plant is a different combination plant. So then you can see many papers people are publishing. They write it there that we are repeating, we are doing this experiment in 2 locations, 3 locations, F2 population. Now do you agree this? Is it scientifically correct? If you think that meaning of replication is that, then it is not correct approach. So that's why I said that in case of QTL, you said effect of each QTL is very small, highly influenced by the environment. So you want to study the effect of the environment. So you need to grow it in many more locations. But your F2 cannot be repeated over many locations. But of course, our rice scientists has developed techniques. What they do? They grow it. When tiller comes, they separate the tillers. Tiller means what? They are the same. So that, I think that case you can do that. So you can have devise different mechanism, that is a different issue. But if you see the overall, that is a different right. Is it okay? You need a mapping population first. Second, what you need? Molecular marker system. Madam, have you got your answer now? That you need a robust molecular marker system if you really want to do the QTL mapping. You need a very good phenotyping facility, right? Because it is highly influenced by the environment. So your data may be biased because not for your faults, but because of the environmental effect, it may be there. So if you want really the precise results, your phenotyping should be very, very accurate, right? Finally, appropriate algorithm and different softwares. So this is available, no problem, but rest of the things you have to manage, okay? All right, let us quickly see the reel. I just want to mention some of the words. You know how to develop the reel. Sorry. The single important thing here is SSD, right? You know about this? What is that SSD? Yes, that is the way you maintain the population. Right? Otherwise, it will go a pyramid. Huge population, you cannot manage that one. That's why you are using that one. Fine. another important, when I mentioned that you should have a robust marker system, you should have marker dense maps, marker dense. It means what? Marker should be in a very close proximity over the chromosomes, over the linkage map. Now if you compare these three maps here, three different maps, which one you would prefer? 2B. That it seems that there is almost uniform distribution and it has covered almost the entire chromosome. Now if you see this one, here you see from this marker is 703897 to this marker there is a huge gap of 18.9 centimorgan, and the other one is here. Now somehow if this marker is monomorphic, it means from this part to this part, whole part is markerless. So markerless means no information. You do not know who is lying there, whether any QTL is there or not. You have no information because no markers. One marker was there and that become monomorphic, so information less. Similar is the case here, huge gap. So in that case you lose many information. You have no idea what is there, right? So that's why try to find out that I should have a genome wide coverage, that is uniformly, not in one place huge dense marker, there's a big gap, no markers, not preferable. It should be preferable if it is uniformly distributed. You know the idea why you need uniformly? Okay, fine, that's why. Now let us quickly go for the steps of QTL mapping. The first step, what you do here, selection of the parents and mapping population development. That already you know and you are telling about that. What we say here, the parents, when you select it, it should be diverse. This word is important, diverse. What is the advantage of this diversity? Please, what is the advantage of the diverse parent? Why you need diverse parent? What is your idea? If it is closed, genetically closed, what will be the problem?
If they are diverse in nature, we will get more segregates.
More segregate. What do you think? Why you need a diverse parent to develop mapping population?
You can get transmission segregation of all the parents.
What is your purpose here? Are anything linked with this transgressive segregant here? Or anyway, no problem. Add last one, you please. Why you need? Why you think? This no problem. This is just think. Why you think that I need diverse parents? Okay. If you take genetically close parents, what will happen?
We can get more diverse parents, we can get more genetic variation.
More genetic variation. You get then more wider, more diverse means you have chances polymorphism will be more. Polymorphism means the difference. Because if they're genetically they are close, what will many of the area will be similar in that case. So most of the things will be common and you will not get any polymorphic marker. You will get all monomorphic because they are same, no difference. So if they are genetically diverse, possibilities are there, I will get more polymorphic mark. So if you take one cultivated type and one wild type, chances of getting this polymorphic markers is more in case of that. Okay, so that's why we said that you need the okay, and we said that why we say preferred real, that is because it is homozygous line and it can be repeated over locations and over years. The first thing, you have got parents and you have developed the mapping population. Now what you will do? You will do the phenotyping, or some people say I will do parental polymorphism first. So these two steps you can do alternative, no problem at all. You can do phenotyping, all right, phenotyping as you know, but we should take care that error should be less and you can take care of the environmental variations. So we say that it should be a replicated one, it should be preferably over locations and preferably over years. This year, if you see current year, in central India, huge rains were going on. So whatever experiments conducted this year, definitely they are not going to get the expected results because of this excess rain. But that data is also important. Next year you grow it, you do not know what is going to be the climatic situations. So you take data also. Okay. So then you finally come to a conclusion, and that will be your more reliable one. That's why we say that you should take care of these things. Replicated trials over years, over the location. Yeah. Then parental polymorphism survey, we already have discussed about this thing. Marker should cover entire genome, right, uniformly, densely. Okay. Diverse the parents, more would be the polymorphism. So you have selected parents, then develop mapping population, you have done the phenotyping now, you have done the parental polymorphism survey, and you have got the markers. Okay. So what with that marker, what you will do? You will do the genotype. As you remember the first table, you need yield data. Say for example yield, there is a quantitative character. Other side you need the marker genotype, capital A, small a, small a. So how you will get that data? You will get that data with those markers through the genotyping. Okay. So each and every plant you have to do that. But here when you do the scoring, please take care of the requirement of the softwares. Okay. Some software they need that data as P1 is equal to 1, P2 is equal to 3, hybrid is 2, and some other software P1 is AA, P2 is BB, hybrid is H, like that way. So accordingly you have to score the data, right? Then you can do for analyzing. This is one way how you are, is about this AA. So this is my P1, is this band, upper band, I am calling it A allele. The lower band is from P2, that is my B allele. F1 is both alleles together, so it is a heterozygous, and rest you can compare and score. This is like A, so A, this is heterozygous, and so on. So this way we do the things. Alright, this is the way you score it. Then finally you compare, compile all those information. Say for marker 1, these are my plant numbers. For marker 1, this is A, this is B. For marker 2, this way you develop, because you are talking about the whole genome. You are covering the entire genome for this genotyping purpose. Each and every plant you are doing. Okay. So finally you get a table like this, just representative table only. Then with these two, you have to find out the linkage map construction, right? Phenotypic data, genotypic data, now you have it, then you develop a linkage map with those data, right? So in that map, what information we get? In that linkage map, we get the information about the order of the markers and in the chromosome, right? What is the order? Who is first, who is second, who is third, like that one. Second information you would get is the genetic distance, right? If you see our previous tables, our previous map, you will get, yes, these are the order of the markers, these are the genetic distance. Say for these two first markers, you are having 6.8 centimorgan, the second pair there is a 3.0 centimorgan, and so on. So you have the order, and also you have got the distance between the markers. Now while doing so, quite often we found that number of chromosomes showing to be more than what is expected. In case of rice, how many chromosomes you should have? Any doubt? Any doubt about 12? No doubt. Okay. But remember that is for n. N is equal to 12, right? Okay. All right. So when you really develop the linkage map, sometimes it may be 14, 15. It may happen so, right? Because what are the data you are giving, with that permutation combination, it is preparing the linkage map. In certain situation, it may go up. What you will do now? In that, to avoid that situation, you add some more markers and do that again, repeat the things. You repeat it until you get exact number of the chromosomes in this map and actual number of the chromosome in the particular crop. Okay? This is easy, you can do it, no problem. Then finally, you do the marker trait association. As we said, the first capital A one group, small a one group, okay. Then you take the yield data, do the statistical significance, then you get, oh yes, this marker is associated with this QTL, this marker is associated with this QTL, and this marker is not associated. So that is called marker trait association. All right. And during that, what you will get? You will get the genetic association. You can locate the QTL where it is, okay, with respect to that marker and the chromosome number. And also this information is important for us. That is your PVE, phenotypic variation explained by that particular QTL. As you said that my character is controlled by many genes. Say for example, 20. Each and every QTL are contributing. We say theoretically they are equal, right? But in reality, they may not be equal. Some may be giving out of 100, some is giving 20, some is giving 5, some is 15, and so on. So that's why you have to estimate that how much contribution a particular QTL is giving to the total variation, right? Total is 100, so how much it's giving, okay? So here you have this marker data, that's your yield data, then you, what we say that marker trait association. So once you map those QTLs, that yes, these are the QTLs I have got. But as QTL mapping is highly influenced by those environment and everything, you need to test your data. Again, that is called validation. Validation. That means what you have got in your experiment, you should test that yes, I have seen that there is a QTL in chromosome number 1. In my population only, because I have used a particular population, mapping population. Now I have to use a different mapping population, or I can use a different population germplasms. So in that case also, again I will see that what QTL I have mapped here, whether or not I am getting it in other population, right? That is called your validation. Once you get it, then you are certain that what I am getting is perfectly correct, and that is usable. That is usable, because many a times you will see some of the cases false positive, that somehow you are getting it because of those some factors, but in reality it may not be possible, it may not be there, right? So that's why you need to validate and check it whether this is. That's why we also do one thing. Why we said that we repeat the experiment over years and over location. Every year we map. So in this year I have got 5 QTLs. Next year again I will map. I will see whether same 5 QTLs are coming or not. Not necessarily all will come. Maybe we get some new or some old. So what old are repeating, we call them consistent QTL. Consistent. So consistently they are coming over the years or over the location. When you get a QTL consistently, you can be sure that this is not a false positive. This is a real one. This is another kind of test that yes, since I am getting it repeatedly over years or over location, it means this is really due to genetic factors, not by mistake or anything, right? So that's why we repeat the same thing over year and location also. How you do that? The process now you know your requirement, what are the requirement you have, that yes, I need population, I need markers, I need your molecular maps and so on things, softwares. Finally, you have done the mapping business, starting from selecting the parents, genotyping, phenotyping, marker trait association, and even your mapping and finally validation. This business is over. Now actually there are certain formula we use or approach we follow. I am just mentioning those, some of those which are basic in nature, right? If you want to map a single QTL, then you have two different types of approaches: single marker analysis or simple interval mapping. If you want to map many QTLs, multiple QTL, so you have a list. There are many others: composite interval, inclusive composite interval, joint inclusive, multiple interval, Bayesian. So many approaches are there, right? I am not going to discuss each and every one, just mentioning few important points. I think Dr. Vinod will take this business again in detail. We will do some practical perhaps, I think so. All right, now let us see some of the things. In case of single marker analysis, what you do here? Single marker, remember, you are now not thinking about all the markers in the map. You are just talking about a single marker at a time, right? You remember the principle. What principle we have discussed? That based on the genotype of that principle, the marker, you divide it into groups, right? So then you see that here you have the mapping population. Okay, you're right. Thank you. Yes. So you have this map polymorph. These are all the things. You have got trade, then you need to do this thing. Say this is this A and small a. This you are talking about, this your QTL, target QTL, and this is your marker. So now you want to see whether this marker M is associated with this QTL, which is represented by A and A. So you take the one marker and see that significance test as we have done earlier, right? Then if you see their variation is there, then you can say this is, say for example, if you this is F2, that's why you have 3 classes. Here you see this is capital M, this is small m, this is small m, capital M. So these 3 genotypes you have. But if you see that statistical significance, there is no statistical significant differences. So you can say that marker is not linked. But if you see this type of variation, which really signifies there is the significant differences, then you can say, oh, this marker is linked with that particular QTL, right? So you take one marker at a time. This is the simplest way. And for this matter, you need not to have any specific software even. Your simple software, it will work. You can say this is very quick, does not need complex software. Even you do not need the linkage map, because you do not know even the position where it is. But where is the marker? You know that yes, this is linked. But problem is it may not be very accurate. Actual position is not known, detection power is less, and here, as I told you earlier, false positive. False positive is called type 1 error. What is type 2 error? Type 2 error. False positive is what error? What that mean? Type 1 error, what that mean actually? Meaning, just internal meaning in very common words.
It shows significant or true result, but we reject it. Then it's type 1 error. In type 2 error, it's actually wrong, but we accept it as a right.
Okay, this is your opinion. Anybody can counter him what he is telling? Or simple words, false positive, false negative. False positive is you are calling it as a type 1 error. False negative is? You tell me. Yeah. No, you only. Yeah. You don't know, it's okay. You know what is false positive and false negative? In simple word, what is false positive? Is it real or unreal? This is unreal. Means the association is not there. You are calling it as an association, right? It's positive. You are calling it as a positive, but it is not positive. It's not unreal, not real. This one, it's a false. So you are assigning that one. Then in the, what is the reverse? Means association was actually there, but you are rejecting it. Somehow you are calling it, oh, there is no association. Now out of this, which one is more dangerous? House is divided. House is divided. Should you go for voting? What do you think, Amresh? False positive is, somebody is not there, you are calling it, what is the wrong? But somebody was there, you are rejecting it. Now which, I don't know, you tell me. What is your opinion? Which one is more dangerous? Dangerous. Type 2. Why? Why type 2 is more dangerous? Both are dangerous.
Sir, in type 1 error, we are validating it on another population.
So what?
But if we skip it all, we cannot validate it in further generation or further population.
If you validate next generation, you will not get it because it is false. It was not real. By mistake, you are calling it as a QTL. It was not there. That's false positive.
False positive.
But it was there, but you are rejecting it. So you validate, you get it.
No, sir. In first, during the validation, we can understand that there is no association. But if firstly we reject it, we don't go further for the QTL identity.
Think about this. I am leaving it to you. I will come to the last day again, just to ask this question. Okay? Which one is more dangerous? False positive and false negative. Okay? I said house is divided, so we cannot take a decision. So we have to wait and giving you a chance to think over it. I'll come back to ask only this question. Okay? And one more. Previous question also. Okay? Note it down. We have to have opinion. Fine. So quickly go to the next. Single marker. This is your simple interval mapping. Simple interval mapping. Okay? Here you do what? Here you are talking about interval. Means you must have two markers. Then within these two markers, you are assuming that there is a QTL. In any place within this, marker, it may be close to M1 or maybe close to this side also, you do not know. But it is there somewhere, right? So you have to validate that one. So for that marker, what you take advantage of this software, LOD, LOD, right? Logarithm of odds. So what is odds basically? Odd is basically this is the ratio. You have to take the data, okay, assuming that there is a QTL and there is no QTL. That is the ratio. That ratio is called odd. What that value comes out from that odd, take logarithm of that. Okay? Say from here you are getting one value x. Take log of x. What is the log of x coming out? See, that is the value. That is the LOD value. Logarithm of odds. First you have to calculate odds. Odds is what? Odds is a ratio of present QTL, absent. If it is a present, what is the value? If it is absent, what is that value? You take that ratio. What is that ratio? Take logarithm of that, that is your LOD. And we normally take that LOD should be 3, right? And LOD 3 means what? It's 1,000 times probability is there that it is correct. So if earlier you see, if you see old papers, then they will have not 3 when they are getting for 2 or 2.5. But gradually now you are increasing. If you increase more, your precision is further increased, right? Nowadays we do not stick for a particular LOD. With the mapping population, we calculate a dynamic LOD. We change it with the population. Okay, that's also. But this was earlier followed like this. So here when you get highest LOD, that is the QTL. If you cross the threshold, your threshold is 3. So here you see this QTL is here. So that QTL is located between marker M3 and sorry, M3 and M4. So that is the, you said interval, composite interval, right? So that QTL is located within that interval, right? This is the one. Merits and demerits, there are so many. It's better than SMA, of course, you know that, but demerits also there. Suppose there are two QTL within that same region, within that same interval, two QTLs. So it's difficult to separate them because their effects are coming together. Okay, epistasis is ignored. Then there are certain methods. I'll just mention who take care of this epistasis. I'll just mention about that. Then there is a composite interval mapping. You are talking about one interval, but here you are taking composite interval. Earlier you considered QTL within that two, within the frame of the two marker. You are not bothering what is happening outside that marker interval. But in this case, you are taking that other markers also, the effect of those markers or their other QTLs. Okay, so that's why you are having a better position in this case. So you say that is more precise and effective. All right, so we'll see few of this. Advantage of what? It's you can go for increasing the precision of the mapping, because earlier, as I said, you do not have any data whether it was influenced by other markers or not other QTLs, but here you are taking care. So what is data coming for your QTL is far better or far precise. All right. Disadvantages are also definitely will be there. As I said, combined effect definitely will twist the data. Multiple interval mapping, here you are taking so many QTLs, so many interval together, right? We will not discuss these things. There is the table, you can later see which one is better and why. It's nothing to worry much. But here this is the current method people are using mostly. As I said that in earlier case, almost rest of the things taken care of here also. But one more thing you are taking care, that if there is a interaction or epistasis, this epistasis, earlier models ignored most of the things, but this inclusive composite interval mapping, it is taking care of that. So you can think what is data you coming to your calculation, far precise. It will give you definitely map like this. So here is again, if you take your threshold is here, so you see only one QTL you are getting. This is a location. All right. For that matter, as what I said, after mapping population, markers, and all this thing, you need the softwares. Many softwares are available. Okay, perhaps there will be one demonstration for software also. So in that software practice, then you can find out which. Okay, many of those are freely available. You can use it. Now overall, what is the advantage of that QTL mapping, right? Okay, so you are getting that a genomic region. You are identifying a genomic region which is controlling your quantitative trait, right? And that genomic region is now identified by the flanking markers, right? Like this, she is sitting here, both sides you have the markers. So those flanking markers will be there. It is advantageous or disadvantageous? Why it is advantageous?
In a selection criteria, if we use a single marker, then 5% error chances and 95% probability that we get. And if you trend marker.
Okay, one more thing you just tell me. In most of the mapping studies, we say this is your target gene. Okay, we say we should have marker on either side rather than one side. Which one you prefer? You need marker only in one side or in marker on both sides of the target gene?
So marker on both sides.
What is the advantage of that?
Sir, it is more reliable.
Why?
Sir, because we know the exact position of the gene where it is located.
Okay. What is your opinion about this? You need marker on either side of the target gene?
Yes, sir, for easy identification.
For?
Easy identify, we can easily identify that particular gene.
What is your opinion about that? You need marker on either side of the gene?
Yes, sir.
Why?
So, to identify the need.
Any other purpose? Not IRS student, other? You, madam? What is your opinion? You don't know, it's okay. No, don't know is very good. You will learn it. Yeah, you said already. Yeah, what is your opinion?
Use it for grading purposes, like we can improvise this QTL.
That is okay, but why? If I have on one side, what is the problem?
We'll assure the complete transfer or
That is, can you explain that thing in scientific, in genetics term? Yeah, please. In terms of recombination, yeah. Why?
If plant gene is on both sides,
Huh.
recombination chance is less, but if it is only one side.
Yes, that is somewhere like this, something like that. Is it? Explain it properly.
Sir, if there is only one marker,
Oh.
then there is a chance of crossing over between marker and trait of
Right?
So we can't identify the target rate.
Okay, okay. What she is explaining, suppose you have the marker here, this is your target gene, you have a marker here, and this is a 5 centimorgan, okay, 5 centimorgan away. How many false positive will be here? Last one. I have 5 centimorgan away. How many false positive will be here? Tentatively. 5 out of 5%. 5%, right? Okay, I have another marker this side that is about 7 centimorgan away. How many false positive? If I take together, how many? Only 0.5? Is it less or more? That is the advantage. If you take both there, the false positive drastically go down. That is the important. Okay, so that's why the breeder will be using those flanking markers and all this thing. Okay, why you need for fine mapping? Fine mapping means you are this mapping of the QTLs. You are going very closer, right? When you go very closer, you can even identify. I told you that genomic region may contain more than one gene. When you are going closer and closer and closer, you can even go to the specific gene for that, right? That's why you need a fine. I will show you one diagram here. Okay. Here you see, just an example. Grain width in case of rice. It was mapped here. How many is the, what is the distance? 10.6, a huge, right? Now what you are doing, this is your clone, these are what, when you do the physical map, right? Okay, after that you are coming very closer, you are coming very close, you are finding markers very closer to the target gene, or even you can, if possible, later, the target nucleotide. Ultimately you will find functional nucleotide, who actually has got changed, because of which one variety is resistant, one is susceptible. Can you do that? So that's why you are going for fine mapping, to know the exact gene and its function. Gene first, then its function. All right, okay. What is the disadvantage? Problem is also there. What is the problem? You said the variation will be limited between the two parents used. Now you have the answer. Somebody else has mentioned that I will go for multi-parent, because your variation is limited to the two parent I used. I can use many parents now, so then problem will be over. You said I have only 2 alleles because 2 parents. Now if you go for many parents, you may get, all right, resolution is maybe low, you need to advance it. How you know that? So as I said, that it may contain many genes together. Is a problem to identify those things. This is another problem, perennial crops. One of my friend is working on mango. Today he will grow one mango. After 7 years it's come to flower. Then he will make cross, get one mango, again do it. When he will get the F2 population, by that time he will retire. Right? So I think scientists are working on that and they have got the solution as well. Can you mention any method where you need not to wait for this? Any other method, any other approach where you need not to make this mapping population, but still you can go for mapping? Association mapping. You need not to develop the mapping population. So solution will be there, don't worry. They said if I go for validation, again I need to waste time and energy, so problem will be there. Anyway friends, we have to close it, but quickly, what are the very important points, because going back you are going to start your work. So before that you have to take care of those things. So what are the very critical points you should be careful? Phenotyping, right? All right. So as we discussed over the, you have to do the experiment over the years, over the location. Size of the population is a very, very important factor. I think I have forgot, yeah, mentioned Bevis effect. Bevis effect. You know what is that Bevis effect? If your size of the population is small, very small QTLs, means QTLs with very small effect will not be detected, and whatever detected, they will have exaggerated effect. Means small QTL, means QTL with small effect will not be detected, and whatever QTL will be detected, they will have exaggerated. Okay, so that is your overestimation will be there. You need marker system. I have the again answer to this. I have another slide to counter all these three things. We'll see it. But anyway, at present you must have, you must understand. Yeah, marker segregation also there. You said marker density should be high, scoring I told you we take care of those. These are the points you should be careful about. How to score, how to take the data, phenotyping, all right. But you are in a post-genomic era, so you see the things in a different perspective. This is what we have discussed in my generation. So those are the take care of those things. But these are the points I said: phenotyping, size, population, molecular marker system. But you are in a different era now. You have many more technological advantages. You have many more understanding of the molecular biological tools and techniques. So they are giving answer to many of those problems. You are talking about, you see, total concept of this QTL mapping is changing. Earlier you said whole organism, one plant is tall, one plant is short, whole plant you are measuring, right? One plant is come to flowering in 45 days, one in 30 days. You take the whole plant data. But now you are forgetting about that. You are going that how many transcripts are developed. How many transcript who is now expressing that, you are talking about that expression QTL. How much protein is estimated, developed. You are forgetting the whole plants. You are going at a particular character at a time to estimate the QTL. So you are forgetting the whole plant, you are taking how much transcripts, how much metabolites, how much protein, right? So those phenotyping you are now coming down, all right? You said I need marker dense. Why you think about that? You have whole genome. Most of the crops you are working now, whole genome is available with you. Why you are worried about those marker system? SNPs are there, thousands, abundant SNPs are there. So why you are worried about? All right, phenotyping. You said phenotyping is a problem because I need to grow Tamil Nadu, I need to grow Srinagar, I need to go Delhi. You are talking about care through this proteomes. All right, you are taking about take care of this. You have the phenotyping facilities now. Phenomics facility you can visit. So you have control environment, phytotron facilities are there. So you are eliminating or reducing the errors. Phenotyping problem is you are taking care through these technological advantages. Now you have other approaches, QTL mapping. Now you are going even to the genome-wide association mapping. So what they are doing now, what is the advantage or advancement? QTL is targeting about the genomic region. You talk about genomic region. But in GWAS, you are going deeper, closer to the small gene itself, or even to the nucleotide, even to the specific nucleotide which was responsible for that change, right? So now perhaps if you do this together, QTL plus GWAS, you can find out the gene directly or even the functional nucleotides. So that's why I told you the future is a different one. All right. it's just one slide away from the thank you. Sub-1 QTL is a brilliant example how that QTL identified, how it was deployed, where you are getting a huge success. But if you see the period, you will be surprised. It took 60 years. They started 2000, in 1950. They got result 2010 here. It's a ERI slide. Okay, sub-1 QTL. It was identified in genotypes collected from Orissa, FR13A, FR13A. From that identified and deployed, now you have seen different varieties with the Qt sub-1, Swarna sub-1, Samba sub-1, everything is there. So now success is there, but you see 60 years of research, not 1 day or 2 days, right? Now from 1960 to 2010, whole years they did the same thing. Finally they got it. I remember one case from Assam. Even before releasing this one, sub-1 QTL variety, they developed locally. Already variety was released, but that was missing from the science. They did not know which QTL or which gene is affecting. What they knew, that this genotype is resistant. So they transferred the character from one gene to the another, one genotype to the another, and finally they released varieties. But they did not know about the QTL. But this was done in ERI. So that was the difference of application of science. They did the same thing, but they did not know the science behind that. And they did the things with science. So you have to apply your mind there, not just doing the things. As you know, that all the important characters are controlled by QTL. So you have to take all care so that we can not only identify, but we can deploy. And the success story of sub-1 QTL and other QTLs, it shows that yes, there is bright sunshine. Okay, thank you very much.
Thank you, sir. Thank you very much. Friends, I'm sure you would have appreciated this lecture. This is Qtl Mapping, which is the complex topic, but it is presented in the most simplest form. So I thank Dr. Talukdar sir wholeheartedly, and I request Dr. Vinod